Yes, J&J 4528 is a second-generation CAR-T product. It differentiates itself from some of the other BCMA CAR-Ts in that it has two BCMA-targeting single-chain antibodies. And these are thought to confer avidity. Basically, you're binding BCMA at two different sites, hopefully for less BCMA escape. It's identical to the CAR construct used in the LEGEND-2 study. The J&J 4528 is actually a confirmatory study of the initial data that was presented in the LEGEND-2 trial. I'm just going to give you a little bit of brief background on that. Is, if you recall, that was a trial that that showed really impressive results at this last ASH with median PFS of 20 months. But one of the concerns has been that the patient population may not have been as heavily pretreated. And maybe the transparency of the data coming out of China was not there. And so the thought was to then actually create J&J 4528 and create this study, CARTITUDE-1, to using the doses used in LEGEND-2 to see if that dose is truly the optimal dose, and then to proceed with the phase 2 study. So this CARTITUDE-1 is a phase 1b/2 study. And here we're presenting the data of the 29 patients enrolled in the phase 1b study. The 29 patients enrolled were heavily pretreated, median prior lines, 5 lines of therapy, 86% triple refractory. These patients underwent the, what now is established, the usual CAR-T schedule of collection, manufacturing of the CAR product, and then lymphodepletion with fludarabine cyclophosphamide, followed by the infusion of J&J 4528. Basically, what we saw was that there's no unexpected safety signals. Virtually all patients had cytokine release syndrome, but mostly grade 1-2. The median time to onset of CRS was longer than what's been described with other BCMA CAR-Ts at 7 days. There was very little neurotoxicity, and only one patient had grade 3 event or higher. Cytopenias were common as expected, but virtually all resolved by 60 days. And there were no significant increase in infectious complications that would be unexpected for this patient population. 4528 was very effective, 100% overall response rate, 86% stringent complete remissions, 97% greater than or equal to VGPR at a median follow-up of 11.5 months. So, responses that we usually see in the frontline setting. 16 patients had complete remissions and had samples evaluable for MRD. And of those, 81% were MRD negative at 10 to the minus 5 and 69% at 10 to the minus 6. The estimated PFS at 9 months was 86%, so quite effective.